Okay. So the question is, if you have cells that are dividing, then they divide again and again and again, you're going to end up with a certain type of growth that we kind of hit on every now and then. And here, what type of growth is that? Exponential. So hopefully I see a bunch of exponential graphs here that you drew me. Yeah, there's one. That's kind of exponential. There's a nice curve shape, Jay. Look at that. Oh, yeah. Now I'm waiting for Family Guy to come get in the fight with the big chicken. All right. Spider-Man? Okay. I have no idea where you guys' minds go on these. I'm worried about our future. Yeah, sure. Did you know why the turkey wouldn't cross the road? You know why anybody think he was a chicken? Sorry. Chicken joke of the day. So we're going to be looking at growth and what happens when you can't control growth. Because we just saw in those exponential graphs that uncontrolled exponential growth of our cells might not be the best thing. So we'll look at the factors that control cell growth and division, certain checkpoints. We'll talk about feedback signals that happen during the cell cycle. And we'll look at cancer and how that's related to the cell cycle and when you can't control it. Yes? Yes, or you'll die. Yeah. Oh, it was good. That was it. That's what I was looking for. Initially. You know, anything. There's not anything that can go on exponentially forever, right? So looking at cell division, we know it has to be highly controlled. You would not want, everybody feel your earlobes. Feel how soft they are, okay? Imagine if your earlobes divided at the same rate. Now feel the calluses on your hands. Imagine if, think about the bottoms of your feet. What if your earlobes were like as rough as the bottom of your feet? You know. So you wouldn't want the skin here to divide at the same rate as the skin on the bottom of your feet, because your ears would be like giant calluses, right, or big old gobs of skin. Okay? And so there must be something that controls the rate of division. That's kind of what we're talking about. So cell growth and division depends on protein signals and other environmental signals. Many proteins within the cell control the phases of the cell cycle. You should all have your notes out. We're on section 3. They got to be somewhere at this point in the game. Okay? So signals from surrounding cells or even from other organs can also regulate cell growth and division. Environmental conditions, including the availability of nutrients, also affect the cell cycle. So I'm guessing that an Ethiopian in Africa's skin divides a little slower than us in America for several reasons, right? Nutrition being one. They got more food than us, or we've got more food than them. Their cells probably are starving, okay? And if I look at the cold weather, your cells divide more in the cold weather. So if I look at just that heat factor alone in the environment, that would also influence the rate of division. So that's environment and nutrients affecting the cell growth rate. Yes? We do. Because we have more nutrients. They are starving. Oh, cold because you need more skin layers. Yeah, that's why you actually grow more hair, nails, and skin in the wintertime. So during the cell cycle, a cell undergoes an inspection process to ensure that the cell is ready for the next phase in the cell cycle. Feedback signals at what are called checkpoints in the cell cycle can delay or trigger the next phase of the cycle. And there are 3 main checkpoints: the G1, G2, and the mitosis checkpoint. How many checkpoints are there? Everybody say 3. Okay. Say G1, G2, mitotic. We'll talk more about G0 later. So looking up here, this is where you will find the checkpoints. We understand that this blue part is interphase, and there will be a checkpoint at the end of G1 before you divide your DNA. And then at the end of G2, there will be another checkpoint before you go into dividing your nucleus. And then in the middle of dividing your chromatids, there will be a checkpoint. Let me ask you this. Where in mitosis would you perhaps stop to check everything? Where do you think? What part of mitosis would you stop and check everything? What would make sense? We know we have prophase, metaphase, anaphase, and telophase. Which portion of mitosis would you stop to check to make sure everything is in order? You know what the cell cycle's favorite? No, that's a bad joke. I'm not telling that one. That's bad. Yeah. They play checkers. That's a bad game. I told you it's bad. It's stupid. I stopped myself. That was like extra bad. Telophase? No. What? Telophase would be too late, right? Everything's divided already. When would everything be organized and easy to check? Everything lined up down the middle, right? Metaphase? Yeah. Anaphase, they're already apart. Prophase, it's all chaotic. That'd be tough, okay? So the first checkpoint, let's talk about these different checkpoints. Before the cell copies its DNA, the cell checks its surroundings. If conditions are favorable and the cell is healthy and large enough, the cell enters the synthesis phase. If conditions are not favorable, the cell goes into a G0 resting period. And certain cells, such as some nerve and muscle cells, stay in that resting period for a long time because they don't divide very often, if at all. We call it the G0, the resting stage, called G0. The second stage, which happens at the end of G2 phase, is before mitosis begins. The cell checks for any mistakes in the copied DNA. Enzymes correct any mistakes. Before you divide your DNA, would you want to proofread it? Yeah, this is where it happens. This checkpoint ensures that the DNA of the daughter cells will be identical to those of the parent cell, and proteins also double check that the cell is large enough to divide. And if the cell passes the G2 checkpoint, it may begin to divide, and once past that checkpoint, proteins will help trigger mitosis. Let me ask you this. The proteins that help mitosis, what are those proteins? What are those proteins? What are the proteins that help trigger mitosis? We'll give you a hint. They are the material that makes up spindle fibers, centrioles, and centrosomes. Livers are not muscles. Microtubulars. Yeah, kind of. That's microtubules. Oh, there's microtubules. Yes, the cytoskeleton protein called microtubules are what make up these structures. And that's what actually initiates mitosis. So during the metaphase stage of mitosis, down the middle, you guys told me that's the best time to check. Chromosomes line up at the equator, and at this point the cell checks that the chromosomes are properly attached to the spindle fibers. You can imagine a little imaginary guy grabbing each of the spindle fibers, kind of like cables, and just checking them, be like, yep, that one's connected, yep, that one's connected. It's a little checkpoint to make sure that everything's in order. Without this point, the sister chromatids of one or more chromosomes may not be separated properly, and this ensures that the genetic material is distributed equally in both daughter cells. If I accidentally grabbed an extra chromosome and put it in one cell and not the other, it could be something like Down syndrome, Klinefelter's, Huntington's disease, all major diseases, right? Not good things. Yep. Yeah. Yep. That happens. Yep. It's called monosomy. No. You'll have a disease. You'll have a genetic disorder. We learn about all those in biotechnology. Depends on which chromosome and if you have an extra one or you're missing one. Yeah. They aren't all the same. Because they all have genes, thousands of genes that you'll either be missing or have extras of. Here's the crazy thing. You can actually have a whole other, it's called disomy, where you have a whole other set of chromosomes. Imagine no centrioles on one side and dragging all of them over to one side. That's actually a good thing. Then you have an extra set to make up for any faults that you have in the other one. So it can be good, but it's very rare that it happens. It's just a fat bubble at that point. So each year, more than 1 million Americans are diagnosed with cancer, the big C. This is a group of severe and sometimes fatal diseases that are caused by uncontrolled cell growth. That means you have lost the ability to control one or all of those phases of your cell cycle. So uncontrolled cell growth and division can result in masses of cells that invade nearby healthy tissue. What do we call masses of cells that invade nearby healthy tissue? What are those called? We'll talk about that. Five, 4, 3, 2, 1. Come on now. We got like 30 of you in here. Parasite, no. It's a tumor, right? It's not a tumor. It's a tumor. It's not true. It's a rumor. Sorry. So uncontrolled cell growth and division can result in masses of cells called tumors. Preventing or curing cancer requires an understanding of how a healthy person's cell can become cancerous. So thinking about those 3 checkpoints. What if you just said, oh, I don't care if I'm ready to divide. We're going to go ahead and divide our DNA anyway. Does that sound like a good idea? No. What if we said, oh, we don't need to proofread our DNA. Let's just go ahead and divide. Does that sound like a good idea? No. If we just learn metaphase, don't even check and say, oh, just hurry up and separate the chromatids. Does that sound like a good idea? No. And all of those things can lead to uncontrolled cell growth. Big mistakes. So normally a cell responds properly to signals and controls. However, damage to a cell's DNA can cause the cell to respond improperly or stop responding by leaving the cell cycle uncontrolled. It'd be like putting your foot down on the gas pedal and, well, maybe not closing your eyes, but like having the gas pedal break and just stay slammed down to the floor. Yeah, like if you're driving a Honda, right? So the defective cells divide and produce more defective cells, and eventually these cells can form a mass called a tumor. And if it just keeps dividing, it's called exponential growth. And that's why when you walk in, your doctor takes a cell count of your cancer cells and says, we got 1,000 cells in you, and you'll be dead in a month. And they know that, because they know those cells are not controlled, right? And they know they will continue to divide because they're not being put into check by your checkpoints properly. So they do know when you're going to die. I mean, that's how they do. They remember how we measured our cell rate and our division rate with the onion root tips? They do the same thing with you, and then they say, okay, based on that, we understand that at this certain date, you'll have millions, not just thousands, and that's a big problem. Yeah.
So, like, say, let's say one of your cells is like
Not one of your cells. They take a sample. Like they'll take what's called a they cut a chunk out, biopsy. A lot of times. Under a microscope, yep. They go to doctor school, oncologists. Well, there's a machine that does it too. But some they have to count by hand. They do it on what's called, I actually have one. It's a hemopoietic cell counter. It's got a laser-etched grid. It goes 4 different ways. I'll show you one in a minute. So a benign tumor does not spread to other parts of the body and can often be removed by surgery. They go in, cut it out, gone. Benign means it does not spread. It's the good kind of tumor if there is such a thing. A malignant tumor is the bad one. This invades and destroys nearby healthy tissue and organs. I'm going to remove your phone if it's not on Peardeck. Malignant tumors or cancers can break loose from their tissue and in origin and grow throughout the body. This process is called metastasis. If somebody tells you their cancer is metastasizing, that's a bad thing. That means it is spreading. Once a cancer has metastasized, it can become difficult to treat. Stage 1, it's localized. Stage 2, it's spreading from one place to another. Stage 3, it's now spread throughout an entire system and into another. Stage 4, it's in your spinal cord. And Stage 5, you are dead because it's in your brain, right? So those are like the stages. Everybody's heard of stages of cancer, right? Yeah, you can get a brain tumor. Yeah, you can. Yeah. Yes. Usually you die before you get stage 5. Yeah. So some cancers can be treated by using drugs that kill fast-growing cancers. And because drugs are chemicals, they call it chemotherapy, or chemo for short. Even if they cut out your tumor, they often give you a round of chemo just to destroy any cells that might have remained. So some cancers can be treated by surgery to remove the affected organ. And then there's, of course, radiation therapy, which is high-energy rays zapping that particular area to destroy the cancer cells there. Now, there's also this cool thing called CyberKnife. You guys ever heard of CyberKnife? Seen the sign maybe on the side of the road? No? Commercials? So everybody point your pencil at the middle of the room at one point. Here, I'll just pick the point. Everybody point right here. So this is the tumor, tip of my pen. Everybody point your laser at my tumor. Okay? So one laser will go right through, like Mr. Ullman here. It won't hurt him at all. Wouldn't even hurt the tumor. But what if all the lasers cross at one point? What do you think happens there? It burns it, right? That's increased energy at one very localized. So they can actually shoot lasers through your body and from all different directions, and it only becomes really damaging where they come together at. And so they can do surgery from outside and not even cut you open and destroy that tumor. So it's kind of cool like that, right? Yeah, they can do it with all kinds of things, like brain. Often they shrink like brain tumors doing that so that they can actually go in and remove them. So there's a lot of stuff they can do. They got to be careful with the lasers because they destroy other tissue while they're in there too. My father-in-law had that done, 3 months ago, and they ended up destroying his ureter along with the tumor. It's the tube that goes from your kidney down to your bladder. So he was urinating inside of his abdominal cavity for about a month before they figured it out. No, it's inside of him. So now he's got a tube that goes, they actually stabbed a needle into his kidney and then piped a tube, and he's got a bag out here that he has to pee in with that kidney now. Well, it's not the same. The catheter goes up your urethra, your pee hole, and then it goes into a bag. So, yeah. Yeah. They're working on that. They've got it kind of experimental stage of that. So the best way to prevent cancer, obviously, is to avoid things that cause cancer. That includes drugs. Yes, pot is in that category, along with cigarettes, alcohol. Any drug is a carcinogenic. Oh, this includes McDonald's too, right? Okay. McDonald's increases free radicals, which are cancer-causing agents. So yes, bad food habits is in this group. Hydrogenated oils. You know, a diet high in processed food. A tanning bed. Who as parent out there has signed off so they can go to the tanner? So, now if they didn't know, that's fine, but that would be like me saying, hey, is it okay if I give your kids some mutated cells so they might get cancer? And you're like, oh, yeah, sure, what the heck, go ahead. But that's essentially what they're signing off on. That should be just as illegal as a cigarette. It's carcinogenic. It causes cancer, for crying out loud. You should be 18 before you have that decision made for you. So they should. Can you imagine somebody like smoking, drinking, eating McDonald's in a tanning bed? Oh, my good Lord. It's like guaranteed death right there. So this does include ultraviolet radiation and sunlight that can damage genes that control the cell cycle. Folks, I've heard the stupid saying that, oh, the real sun is just as bad or worse than the tanning bed. Not true. Okay, the tanning bed is a full day's worth of sun in about a 10-minute period. Come on. A whole day's worth of mutations in 10 minutes. Come on now. We're smarter than that, aren't we? Okay. So chemicals in cigarette smoke also affect cell growth and division and how it's regulated. And then, of course, I hear the one, pot doesn't cause cancer. And I look at them and I say, yeah, it probably causes cancer more. It doesn't even have a freaking filter on it. Okay? It's probably even worse for you. It's like sticking your head over a campfire. Like, who does that? And just inhale. No. No. Carcinogenic. Okay, same thing as a grill. No one sticks their head in their grill at home and just inhales the charcoal, or goes up to the exhaust of a car and says, give me more of that. You know, no one does that. Well, unless they want to die. So, I think you get the idea. By the way, if I see any of you smoking cigarettes, I don't care how old you were, I will come up, grab your cigarette out of your mouth, and wrinkle up your pack and throw it away. I've done it to a 30-year-old man that I had as a student before. He wasn't too happy, but he looked at me and he laughed and he said, you said you were going to do that. And I did it. I do it about once a year.
You're like 30.
I don't care.
No, like how could he be 30 if you're like 30?
No, he looked 30. He probably wasn't 30. He was probably like 25, something like that.
I was just saying, like, you know, like.
I've got students that are now coming back to teach. It's a little creepy. Yeah, I know. So cell growth and division depend on protein signals and other environmental signals. So how your cells divide and grow depends on the environment. You put them both internally and externally. Feedback signals at key checkpoints, G1, G2, and mitotic checkpoint can delay or trigger the next phase of the cell cycle. And uncontrolled cell growth and division results in tumors, cancer, that can invade surrounding tissues and cause the disease noted as cancer. All right. Other than